Welcome to the first in Tecan's 2013 webinar series. I'm Ashley, the Managing Editor for Technology Networks, and I'll be moderating this event. Tecan is introducing this series to help researchers stay informed of the latest techniques to help advance their work. Cutting-edge biological applications, including cell models for prolonged pharmacokinetic studies and 3D cell systems to mimic in vivo situations, as well as increasing the performance of your cell signaling biochemical assays, will be presented. Today's webinar, entitled Helping You to Shape In Vitro Models with Cell-Based Assays, will focus on cell-based assays and how to overcome some hurdles usually associated with cell biology research, in particular with long-term studies. Here, Tecan will introduce ingenious solutions to increase performance and productivity in the lab for cell-based in vitro models. The presentation will be given by Christian Oberdanner, who's a Marketing Application Specialist in Detection for Tecan Austria. Following the webinar, we'll have a Q&A session, and I'd welcome any questions that you may have. To ask a question, you should click the Ask a Question button on the right of your screen, enter your email address and question, and click OK. We will attempt to answer as many questions as possible during this time. If you experience any technical issues, please use this button to request support. I will now hand over to Christian for the webinar.
Thank you, Ashley, for that very nice introduction, and a warm welcome to the audience from my side. As Ashley has already mentioned in his introduction, this webinar today will be about cell-based application, and I want to focus on features of our instrument, which are microplate washers and multimodular microplate readers, and how these features can help for better cell handling to gain optimized results. Looking into the past, but also in the future, we see a dramatic increase of cell-based assays in all areas of life science research, academia, biotech, and pharma. And the overall global market focus for cell-based assays is expected to be around $15 billion in 2015. Taking a closer look to the area of multimode microplate readers, we see that the application area which is most interesting for multimodular microplate reader users is, again, cell biology. So why is there such a general trend to cell-based assays? The answer to this question is pretty simple. The results generated with cell-based assays are of a higher biological relevance than those generated with biochemical assays. This is simply due to the fact that cell-based assays provide a better indication of the in vivo situation than a standard biochemical assay. This is especially important for drug development, because collecting information on how drug candidates interact with cells early on in the drug discovery process will help to identify the prime candidate and will help to avoid late-stage failures. So exactly this in vivo modeling in the early preclinical stage of the drug development process is driving the demand for cell-based assays. As identifying crucial safety and efficiency data will save tens of millions of dollars for the pharmaceutical companies per year. So talking about cell-based assays, there is another trend which is worth mentioning. And this is simply the trend to live cell assays. So researchers are looking for smarter decisions sooner. They want to have more biological relevance in their data, and therefore cell-based assays are now developed by using living cells rather than a cell lysate. This is beneficial because you can monitor cell responses in a time-dependent manner, which facilitates the investigation of a biological process. So this is more or less endpoint data versus kinetic data. So the trend goes from biochemical assays to model organisms. And today I want to focus on cell-based assays with living cells. Of course, Tecan instruments can be used for all 4 categories. So especially our multimodular microplate readers have been successfully used for biochemical assays, also for cell-based assaying using cell lysates. Of course, cell-based assays with living cells, where I want to focus on today. But we also have success stories where customers perform their assays inside a model organism and use our microplate readers for signal detection. So what is the typical workflow of a microplate assay with living cells? First of all, you need to seed the cells in the microplate and let them grow at cell-specific conditions until a certain confluency is reached. After that, most of the workflows have a washing step. So the cells need to be washed, the cell culture medium needs to be exchanged. And for this step, of course, our Tecan microplate washers can be used. And what the benefits are, I will explain later on. After that, there is a treatment of the cells. Most of the time, the treatment is done by adding a compound, but you can also set another stimulus. For this step, we provide another product, which is the HB D300 dispenser. Unfortunately, I won't talk about this instrument today, but for all people in the audience who want to know more about this product, I want to invite you to visit our homepage. After the treatment of the cells, there is most of the time again a washing step. So you sometimes need to get rid of the compound, need again to change the cell culture medium, and of course, Tecan washers can be again used for this. After this washing step, you normally do your assay. So you perform the assay procedure, which sometimes again includes washing, and then you do the signal detection. And when talking about cell-based assays with living cells, you rather do a kinetic measurement than an endpoint measurement. And here our multimode readers come into the game, and how they can help you to get more relevant results, I will explain. So some typical applications with cell-based assays using living cells. Most common applications in this area are cell proliferation, viability, and cytotoxicity assays, because you always want to know what's happening, not just in a single measurement, endpoint measurement, but over a certain time frame. Also migration assays. Also for migration assays, living cells are used, and for reporter gene assays. This is very common. Another very important application field is microbiological growth monitoring, but also calcium release assays are done with living cells. So let's start with cell washing. I now want to spend a couple of minutes on this topic. When thinking about cell washing in a lab, especially in an academic lab, this is really a daily challenge. Manual cell washing using standard pipettes for both aspiration and dispensing is very tedious and time-consuming. So most of the customers do the aspiration with a vacuum-based approach. So they use a vacuum pump, but this is often much too harsh, and many cells will get lost, and the cell viability of the remaining cells will be affected. And even if you are in the lucky situation to have a plate washer in the lab, you really need to make sure if this plate washer is equipped with a cell washing mode, as standard ELISA washers are again much too harsh in their settings and therefore suboptimal for cell washing. As you can see here on this picture, the manual cell washing really affects the cell layer, and on the picture above you see weakly adherent cells, and then the bottom you see strongly adherent cells. And for both cell lines, you see that after cell washing, part of the cells are simply washed away. So how can a plate washer, better a Tecan plate washer, help to overcome this limitation? We provide a certain cell washing mode, the so-called cell protection mode. And this is a combination of tunable aspiration settings, of a very gentle drop-wise dispensing rate, and of a ingenious move function to minimize turbulences. And this combination results in a very high cell retention rate and in a high viability of the cells that are remaining. This function is available for both of our washers, the HydroFlex washer with an 8 or 16 channel head for low to medium throughput, but also for a high-end HydroSpeed washer with a 96 or 384 channels head for high and ultra-high throughput applications. I now want to share some data with you that we generated during the validation of the cell protection function for the HydroSpeed plate washer. And we showed in this note that we can perfectly wash even very weakly adherent cells. We conducted this study at our partners lab in the University of Salzburg. And we investigated 2 different cell lines. So we used a very strongly adherent cell line, a human epithermoid carcinoma cell line, and a very weakly adherent cell line, a mouse mastocytoma cell line. We cultured them in a 96 well plate and in a 384 well plate, and then investigated the wash efficiency by monitoring the removal of a colored solution. Therefore, we used trypan blue at a certain concentration, measured the absorbance after staining, then washed the cells, and again detected the absorbance at 565 nanometer to detect the remaining trypan blue in the sample. Besides analyzing the wash efficiency, we also wanted to know how many cells are remaining and how viable are the remaining cells. And therefore, we determined the cell viability by using an MTT assay. All the results were then compared with those generated with manual cell washing, so the standard approach that is done in that lab. When we take a look into the microscope, we already see that after cell washing, the cells remain, so there is no significant loss of the cells, whether it's strongly adherent cells or weakly adherent cells. Taking a closer look to the results of the 96 well plate, we see that the wash efficiency is good for both cell lines with the washer and for the manual washing. So we have above 95% wash efficiency in all situations. This is really great. But then, taking a look to the cell viability, we see significant differences. For the strongly adherent cell line, the cell viability after washing with the microplate washer is around 80%, but very shockingly after manual cell washing it's just around 20% that is remaining. That means a lot of cells get washed away, and those who remain are really affected, or the cell viability is really affected. For the weakly adherent cell line, the results are even more dramatic. Just 10% of the cells of the cell viability compared to the control is remaining. So that means a lot of cells get washed away, and again, those that remain are affected in cell viability. This picture gets even more convincingly when doing the experiments in the 384 well plate. So again, the wash efficiency was good in all situations, but the cell viability again was very much affected after the manual cell washing, especially for the very weakly adherent cells, which just 3% of the control. Of course, these results are very dramatic, and it might look a little bit different in other labs or using other cell lines. But basically, this data tell us that using a plate washer for cell washing does not only increase your productiveness, so enables a higher throughput, but it also results in better cell retention rate and in the better cell viability of the cells, and this finally results in more reliable results in a higher biological relevance. So let's come to signal detection. Let's talk about multimode microplate readers and cell-based application. When we look to the reader requirements for cell-based assays, the first thing we need to think about is signal detection. Of course, the multimode reader needs to have all the important detection modes to be able to handle cell-based assays. Cell-based assays are developed for all the different detection technologies. That means some of them are based on absorbance, others on fluorescence, and again others on luminescence. And of course, the microplate reader should have all these modules implemented or the capability to upgrade the instrument with a certain module. Besides this very basic requirement, there are other requirements that are related to signal detection. So the reader should have some special optical features to improve signal detection when it comes up to assays using living cells. And this is mainly due to two reasons. The first one is when working with adherent cells that do not attach homogeneously on the surface. So this is always the situation unless the well is totally confluent. You will face the problem that with a common microplate reader, you will never detect the signal of all of these cells. But there is also a second problem, and this is simply the fact that cell culture media normally contains a pH indicator, phenol red, and this one critically interferes with the optical measurement when doing the fluorescence detection from the top. But there are also other requirements related to cell-based assays with living cells, and this is how to maintain the cell viability and the proliferation of the living cells inside the microplate reader. So the first requirement is, of course, to maintain an optimum temperature for the cells. So the instrument needs to be equipped with a temperature control. When working with microorganisms, bacteria or yeast, then it's very important to have an efficient shaking function, because the microorganisms need to be kept moving in the cell media for a better supply with nutrients. Besides these two very basic requirements, there are also some more advanced requirements, and this is the CO2 and O2 control, which you normally find in a cell incubator. The CO2 control is needed to regulate the CO2 level in order to maintain an optimized pH of the cell media. And the O2 level is important when working with anaerobe or microaerophilic organisms and to mimic conditions like in blood to induce hypoxia or things like that. And last but not least, especially for all the long-term experiments conducted in a microplate reader, there must be a solution to avoid or at least minimize the evaporation of the cell media in the microplate wells. With our Infinity Multimode Reader series, we offer solutions for these problems or fulfill these requirements. The Infinity Multimode Reader series consists out of our mid-range readers, the filter-based Infinity F200 Pro, and the monochromator-based Infinity M200 Pro. But we also offer instruments, high-end instruments with a higher speed and higher sensitivity which are the Infinity F500 and the Infinity M1000 Pro, our flagship instrument. Taking a look at the optical features that we offer with our instruments, we see that we have everything that is needed to measure cell-based assays. So the instruments are packed with the optical detection modes ranging from absorbance over fluorescence, fluorescence polarization, time-resolved fluorescence, but also different kind of luminescence, modules, glow luminescence, flash luminescence, multicolor luminescence, and even alpha screen and alpha laser. The instruments also come along with some additional features, and some of them are needed to conduct cell-based assays. Shaking and heating function I already mentioned. Then the set optimization for Fi top and optimal read for Fi bottom, and I will quickly explain what this is. And then, and here I want to focus on in the rest of this webinar, the gas control module, our environmental control option. So let's quickly explain the optical solutions that we have for cell-based assays. The first one I want to introduce to you is the enhanced fluorescence bottom reading with the unique optimal read function. So I already mentioned, whenever working with non-confluent adherent cell lines, you will face the problem that these cells will not attach homogeneously on the surface of the microplate well. So the cells will grow in islands, and in some situations the cell more tend to grow at the very edges of the microplate well. So in a normal microplate reader, you have a light beam with a certain size. That should fit, of course, in all the different well diameters. So in a 384 well plate, we will cover with a standard light beam most of the well. But the same light beam in a 96 well plate, of course, covers a significant smaller area of the microplate well, and this is even more dramatic, for instance, in an 84 well plate. With our ingenious optimal read function, we are enabled to measure the whole well, no matter which plate format is used. So the software will select the best possible pattern for multiple reads per well, and the flash number that is selected in the software will be splitted by the number of measurement spots, which will result in an almost uncompromised measurement speed. So if you want to learn more on how this function can help to get better results, to improve your data, I want to encourage you to simply download this application note shown at the left lower bottom here of this slide, called Fluorescence Bottom Reading for Cell-Based Assays. The second optical solution we have to improve cell-based assays is the automated set optimization with integrated background correction for fluorescence top measurements. Automated set optimization is available with a lot of different microplate readers on the market. But with Tecan, you have that unique integrated background correction function. And this really helps to choose a set position of the focus of the light beam inside the well that has the best signal to blank ratio. And as samples that contain cell culture media with phenol red always show a very high blank signal, this function really helps to improve such kind of measurement. And again, if you want to know how to exactly use this function, I want to again encourage you to download the application note shown here on this slide, called Improved Fluorescence Top Measurement. So coming to the main topic of this webinar, and this is how to maintain cell viability and cell proliferation in cell-based assays. So especially our Infinity 200 Pro series is packed with a couple of these features that help to improve or to maintain cell viability. I already mentioned the shaking capability and the heating capability. But I want to talk about these two functions as this is really basic and perfectly works with our instrument, which has been demonstrated over the last few years. What is new and what is really exciting is the environmental control we offer with our gas control module. So the Infinity 200 Pro was the first multimode reader on the market with adjustable gas concentration. It was the first fusion of an incubator and a reader. So what can the gas control module do exactly? The gas control module can first of all control the CO2 level inside the reader's measurement chamber in a range from 0 to 10% CO2. But it can also control the O2 level between 0.1 and 21%. This is done by replacing the oxygen with nitrogen gas. With the gas control module, we also offer a dual mode. That simply means that you can control CO2 and O2 levels simultaneously. We also offer a manual mode to monitor O2 and CO2 level of premixed gases, which is important for some certain types of microorganisms. And the module also comes along with additional features like acoustic warnings if gas pressure is not okay, with log files in the software to monitor the gas concentrations over the whole experimental period, and a unique altitude control for a better performance of the gas sensors. So what are the benefits, what are the application-related benefits you have when using a gas control module? I think it's clear for everybody that the regulation of CO2 simply stabilizes the pH value of the cell media, and this then ensures the cell viability and proliferation of the cells, and therefore enables for long-term in vitro studies with eukaryotic cells. This function has already been successfully used by our customers, and the first paper was published last year, and you see the reference down at the very bottom of this slide. Besides this CO2 regulation, the simultaneous CO2 and O2 regulation enables for even more exciting applications. It ensures the cell viability and proliferation of anaerobe and microaerophilic organisms, but you can also use this function to mimic physiological conditions like you find in blood. You can mimic phenomena like ischemia or reperfusion, and you can even induce hypoxia, which is very relevant, for instance, for cancer research. Besides these applications that are now enabled by using the GCM, there are some general advantages that are worth mentioning. When you normally want to do such kind of experiments, especially in an academic lab, you need to transfer the plate from an incubator to the reader for each and every measurement step. That means that this is really time-consuming and tedious. And if you do very long experiments over several days, you even will lose data points because you sometimes really need to get to bed and take a sleep. So by using the GCM, you lower the risk of contamination. You have constant atmospheric conditions over your whole experimental period, especially important when you are working at low O2 levels. You have an exact monitoring of your data, of your growth curves, with no gaps in the overnight data. And finally, this results in a very constant and complete data set that guarantees for high-value results. We have, of course, validated the benefits that we claim for the GCM and generated some nice papers, and I now want to share the data with you. The first thing we did is we validated the capability to maintain cell viability of eukaryotic cells with the GCM. Then we wanted to know if this also comes true for microaerophilic organisms. In a third study, we induced and detected hypoxia using the GCM and the 200 Pro. And finally, one of our partners, Promega, did a very nice experiment using the GCM for real-time cytotoxicity measurement. So in a first study that we conducted in collaboration with Thermo Scientific and with the University of Salzburg, we wanted to find out if the GCM is able to maintain the cell viability of eukaryotic cells. The experimental setup was the following. We used GFP transfected human epithermoid carcinoma cells and cultured these cells in a specific microplate provided by Thermo, the so-called NUNC edge plate. And this innovative plate is equipped with an evaporation barrier that can be filled with liquid or agarose to avoid evaporation of the liquid in the sample wells. And as you will see in the next slide, this perfectly works in combination with our reader. We then cultured the cells for 75 hours at 5% CO2 and 37 degrees Celsius inside our reader. We used two different approaches, so we once used cell culture media containing FCS and cell culture medium not containing FCS. And to detect the cell proliferation, we measured the GFP signal every hour. So let's take a look at the results. In the left bottom of the slide, you see the remaining filling volume of the wells, of the liquid in the wells of the NUNC edge plate. And as you can see here, for most of the wells, the loss or the evaporation is not significant at all. So we have between 90 and 100% of the original filling volume remaining. Only at the very edges of the plate we see a little bit more evaporation, but this is simply due to the fact that after 75 hours, the liquid in the evaporation barrier of the plate already totally drained. But of course, as a workaround, you could refill this evaporation barrier, let's say, every 24 hours and theoretically extend the experimental period even more. On the right side of the slide, you see the cell proliferation of cells cultured with FCS in the cell culture medium or without vidrocalcero. For both situations, we see a pretty similar picture. Just the cells that are cultured in an atmosphere containing 5% CO2, so using the GCM, just these cells keep proliferating over the whole experimental period. Cells without any CO2 regulation, but of course cultured in the NUNC edge plate and at 37 degrees Celsius, they will stop proliferation after an overnight period, and they even will die after that. So this result simply shows us that we really need to have a CO2 control to maintain the cell viability of eukaryotic cells if you want to conduct your experiment inside your reader and without the need of transferring the plate from an incubator to the reader every hour. But does this also come true for microaerophilic organisms? So does the GCM enable growth studies with microaerophilic organisms? And to find this out, we again started a study in collaboration with the microbiology department at the University of Salzburg and again with Thermo Scientific. So we again used the NUNC edge plate to avoid the evaporation, and we monitored the growth of Helicobacter pylori inside the Infinity 200 Pro equipped with the GCM. We therefore set the O2 and CO2 regulation of the instrument to 10% CO2, and we varied the concentration of O2. So we either used no O2 regulation at all, we set the O2 to 10% and to 5%. We did the growth monitoring over a period of 28 hours at 37 degrees. And to keep the bacteria moving inside the liquid for a better supply with nutrients, we did continuous shaking over the whole experimental period. To detect the proliferation of Helicobacter pylori, we did an absorbance measurement at 600 nanometer. So this is the standard approach to detect proliferation of microorganisms in a microplate. But we also measured the GFP signal of the cells, as the cells were also transfected with GFP. So let's take a look at the results. And I think the picture here is pretty clear. On the left data plot, you see proliferation detected by measuring the OD600 absorbance. And on the right side, you see the results when measuring the GFP signal of the cells. And of course, for both, the picture is the same. For cells cultured without any O2 regulation, we do not see any proliferation at all. When setting the O2 level to 10%, we see proliferation, but after a very long incubation period of above 20 hours. So not what we would expect. Only at 5% O2, we see a growth curve that we would expect. Growth of the cells start approximately 3 hours after seeding the cells in the microplate after starting incubation, and we have a linear growth range up to 15, 16 hours. So these results simply show us that only with a gas control module equipped with simultaneous O2 and CO2 regulation, you can conduct growth monitoring with microaerophilic organisms. In a third study, we wanted to know if we can use the GCM also to induce and detect hypoxia. Hypoxia, which is a situation with very low oxygen inside the cell. This is relevant for a lot of pathological conditions like cancer. And we tested the induction of hypoxia by doing 3 different experiments. In a first experiment, we induced the hypoxia-inducible factor, the HIF protein, using the reader with GCM. And as a control, we induced hypoxia also by using a dedicated incubator with a simultaneous CO2 and O2 control. And we did this by using a cell sensor line from Life Technology specific for hypoxia. So this cell sensor line contains a hypoxic responsive element. And the readout of this cell sensor line is gene blazer based, so you will detect a threat signal with the microplate reader. We induced hypoxia for 8 hours at 37 degrees using 5% CO2 and setting the O2 level to either 1% or 0.2%. In a second experiment, we wanted to verify the outcome of the first one and simply used again the reader or the dedicated incubator to induce hypoxia, but then did a non-reader based readout or detection. So we did a common immunoassay, we did a simple Western blot on the HIF protein and the VEGF protein. In a third experiment, we selected a chemotherapeutic drug called tirapazamine, which is only cytotoxic under hypoxic conditions. And we treated HCT cells with this compound and then monitored the cell viability using the Life Technologies Alama Blue assay. So let's take a look at the outcome of this study. The results of the first experiments are very much convincing. So you see here, we can perfectly induce hypoxia using our reader with GCM, and this results in non-significant difference to hypoxia induction using a standard incubator. Of course, both systems are valid to induce hypoxia, as you can see here from the comparison to the normoxia control. This comes true for 1% O2 and even more clear for 0.2% O2. So this first experiment showed, okay, this works, and the nice thing is that by combining all the different functions of the microplate reader, we could automate the whole workflow, as we could simply inject the assay reagent for the gene blazer assay with our injector system. In the second experiment, where we wanted to verify the outcome of the first one, we again induced the hypoxia using the reader or the incubator, and again we see the same picture. So both instruments, the dedicated incubator as well as our reader with GCM, perfectly work to induce hypoxia. So we have the same result when measuring the VEGF protein or the HIF protein. So the outcome of the first experiment was verified with this one, and finally we wanted to see the cytotoxicity of tirapazamine under hypoxic conditions. So we did a dose response curve with tirapazamine using the GCM module and setting the O2 concentration to 1% or to 0.2%. And what we see here is that at 1% O2, we see loss in cell viability, but this is not really dramatic and just visible at very high concentrations of tirapazamine. But then lowering the oxygen concentration to 0.2% shows that tirapazamine is cytotoxic even at lower concentrations. Again, the outcome for the incubator and our reader were pretty much comparable or the same. And the overall outcome of this experiment tells us that hypoxia just starts at 1% O2. So we really need to go down to a very low level, and you really need to have the capability of your instrument to simply control such low oxygen levels. And this is what you can do with the Tecan reader. You can even go down to 0.1% O2. Okay, and finally, in a last study, Promega tested our GCM module and the Infinity 200 Pro for real-time cytotoxicity analyzing using their CellTox screen assay. This non-activity-based cytotoxicity sample works in the way that the dye itself cannot enter viable cells. It cannot pass an intact cell membrane. But when the cell is dying, the membrane gets leaky, the dye can enter the cell and will bind to the DNA. So this is a non-toxic cytotoxicity assay and is therefore perfectly suited to conduct long-term experiments in a real-time manner. Promega used our HPD 300 digital dispenser for dispensing the test compound, and then they used the Infinity 200 Pro with GCM for the kinetic measurement, for the real-time kinetic measurement. And they did this for 72 hours at 37 degrees using 5% CO2 and did a fluorescence measurement every hour. And the results, which they presented in the poster, are again, I think, clear for everybody. Because you can perfectly monitor the cytotoxicity over the whole experimental period with no gaps in the data at all, and therefore the overall quality of the results is much better than just doing endpoint measurements or by transferring the plate from the incubator to the reader for every hour. So Promega is really happy with this, and their assay, the CellTox screen assay, perfectly fits to our instrument. The perfect combination for studying cytotoxicity in a real-time manner. We have also a lot of other stories to tell about cell-based applications with our Infinity multimode readers. I just want to share some slides with you and give you some hints, and maybe you're interested in one or the other note that we offer on our homepage. The first note is a technical note that explains cell proliferation and cell viability assays, focusing on MTT, MTS, and resveratrol-based assays, and how you can optimize the reader to conduct such applications. Another application note is about growth monitoring of bacteria. Here we use Staphylococcus aureus, and we showed how to optimize the system, especially the shaking parameter and the plate format with the filling volume to get best possible results of your growth monitoring. We also have a very nice white paper written by one of our customers about cell migration. And here you will learn how to use the Infinity M200 Pro using BD Falcon FluoPloc microplate. And in this study, the customer perfectly showed that using the instrument is as sensitive as using an inverted fluorescence microscope, but you have the benefit of a significantly increased throughput and a much better statistical relevance. We have a brand new application note together with Life Technologies about their next generation resveratrol-based cell viability assay and how this works on an Infinity M200 Pro with GCM, but also on an Infinity M1000 Pro microplate reader. And finally, we even have a lot of application data available about 3D cell culture applications. And this note here will explain how to analyze biological drug effects in 3D using microtissues generated with the Inspiro system and detected on the Infinity M200 Pro microplate reader. We even have to tell a lot of more, and therefore we have our Tecan journal with a lot of interesting success stories, and you're all invited to download our Tecan journals from the link shown here on this slide. I herewith also want to thank our partners in cell biology, and I want to share with you some important links. So if you are interested in Tecan in general, just visit our homepage, tecan.com. If you are interested in microplate washers and multimodular microplate readers, use the link tecan.com/detection. If you want to learn more about the Infinity 200 Pro, often called the cell-based reader, just use the link tecan.com/Infinity 200 Pro. But we also have some nice application-related landing pages for 3D cell culture, for the GCM, again, showing all the benefits I just presented, and about our hydrospeed plate washer with the cell protection mode. So I herewith want to thank you all for attention, and I'm ready for questions.
Thank you very much, Christian, for the presentation. That was really interesting. I'm sure everyone will agree. It raised some very, very interesting points. So we now enter the Q&A section of the webinar. We've had a number of questions in. I would say at this point, if anyone has any more questions, please continue to submit these, and we will certainly try and get to as many as possible. So the first question I have here, Christian, is exact monitoring of O2 concentration really an issue when doing hypoxic studies?
So, well, like I have tried to explain during the presentation, it's of course very important to have an exact monitoring of oxygen whenever you're working with hypoxia. And it's especially important to be able to go down below 1% with the oxygen control to be able to see all the important events that are related to hypoxia.
Excellent, thank you very much. The next question we've had in here says, does the optimal read function in the Tecan reader, does that influence the reading time at all?
It does to a certain extent, of course, because you have several measurement points inside the well compared to just one. But with our technology, we reach a nearly uncompromised measurement time by splitting the flash numbers to the certain measurement points that are pre-selected by the software. So yes, measurement time is affected, but this is not really significant compared to a single measurement reading.
Okay, excellent. Thank you for that. And finally, the question we have here is it possible to allow multiplexing, so for instance, absorbance fluorescence detection, when doing co-culture assays using different cell lines?
Yes, it is, of course. Our software is very flexible, and you can basically set up every measurement script you could remember or think of. We could show in several different studies that we can do multiplexing, and a very interesting one was published in the December issue, where we showed that we can do fluorescence and luminescence multiplexing in a 3D cell culture environment, even in that. Yeah.
Excellent. Well, thank you very much. And well, that's all the questions we've had in. So I'd like to thank everyone again for attending the webinar. And like I say, I'm sure you agree it was incredibly interesting, and I in particular found it incredibly useful as well. I'd also like to thank Christian for the presentation. I think finally I'd just like to draw everyone's attention to the next Tecan webinar coming up. This is going to be on the 2nd of July, and this one's going to be entitled A Lesson for Cell Signalling Using Advanced Detection Techniques. So we'll obviously be in touch to let you know more about that nearer the time. So again, thanks for attending, and we shall see you for the second webinar in the series. Thank you.
Thank you very much, and goodbye to the audience.